In this video, I'd like to explain one of the major features of this vector that we're cloning into PQE31, and that is this thing right here. It's called a his tag, and I want to explain why that is in the vector and what we're going to use it for. Remember that the most common use for molecular cloning is in the lab. These labs could be biochemistry labs, biology labs, even physics labs sometimes use cloning because they're looking at biophysics. And in all of these cases, we're usually cloning in some gene or some form of a gene into a plasmid, and then we're using that, we're putting that plasmid into different types of cells and expressing that gene. In many cases, that may be because we want to learn what the gene itself does, but in other cases, we may want to use that gene to manipulate the biology of a cell and observe how that manipulation changes the cell's functioning. Perhaps we create a mutation in the gene that is the same mutation that is found in a disease, a genetic disease. And so we mutate the gene to contain that mutation that's disease-causing, and then we put that mutated gene into a plasmid through molecular cloning, and then we put that plasmid into cells and express it. Then we can see how that mutated gene affects cell functioning. Often it's useful and helpful to be able to track this gene that we're expressing in the cells. It may be that we want to test what other proteins this protein that we're expressing interacts with. It may be that we just want to confirm that this gene is being expressed like we wanted it to. In other cases, we might be wanting to test if this gene is upregulated or downregulated in the cell. Perhaps when the protein is expressed from the gene, the cell destroys this protein, or perhaps if it's a mutation, perhaps this mutation in the gene causes the protein to last longer than the unmutated version. But in general, it's useful and helpful to have a way to track that protein and be able to monitor that protein that we're expressing easily in cells. And so in many cases, the way we'll do that is attach a tag to that protein so that we can monitor it. So in some cases, that tag may be a fluorescent tag, and so you can monitor where that protein goes by attaching GFP to it, for example. And then with a microscope, a fluorescent microscope, you can follow where that protein goes, what it does, how much of it is expressed, where it's localized in the cell, etc. But there are other types of tags too that you can attach to proteins, and these tags are very helpful in allowing you to do biochemistry on the protein to see how much of it is expressed, what partners it interacts with, and also where it's localized in the cell. These types of tags that allow you to separate out the protein that you're expressing from all of the other proteins in the cell are called affinity tags. So let's imagine that you express this protein in a cell and you let it interact with whatever it's going to interact with. It interacts with some sort of binding partner. And to understand the biology of the cell better, you want to know what partner it interacts with. So you don't know what this partner is. Well, one way that you could do that is you could pull this protein out of the cell and then using biochemistry figure out what is bound to it, because presumably if you break open the cell and remove all the proteins and you remove this protein, if in the cell it was attached to this partner, then when you pull it out in a tube and look at it with biochemistry, if you've treated the cell correctly, that binding partner should still be attached. And then using biochemistry, you can identify what that partner is. This can teach you a lot about how proteins interact in the cell, which is really how life happens, right? Is how all these proteins interact with each other in the cell. So people are very interested in what partners there are. So how can you if you have this cell with all sorts of proteins expressed in it, including the protein that you want to know about, that you've cloned into the plasmid here, how do you separate that protein out and the partner that it's bound to and exclude all the other proteins that are in the cell so that you don't accidentally look at them and get a mixed message of what's bound to what? Well, these affinity tags are pieces of, they're typically amino acids or small molecular uh, moieties, which is basically a structure that you can attach to these proteins, and these tags act kind of like Velcro on the protein. They make the protein sticky in a way so that you can flow the proteins of the cell across a column that's just sticky for this tag. And so as the proteins flow across this column, only the proteins that have the tag on them will be sticky and stick to that column, and all the other proteins will flow through. So you have this column, and you break apart the cell here. So you'd break apart the membrane, and you'd release all the protein to create what's called a lysate. And with that protein lysate, you can add it to this column, and this column has some sort of bead in here that sticks to this affinity tag, so it has a lot of affinity for it. When you then add when you add the lysate from the cell to this column and allow things to drip through, anything that doesn't have the tag will just drip through. And that would be all the other proteins except for this one protein that you've cloned that has this his tag, this affinity tag. And there's various types of tags. His is one of them. But that allows you to separate out the proteins that don't have the tag from this protein, this one protein that does, and whatever binding partner it's bound to. Then you can elute, after you've washed all of these other unbound proteins away, you can elute your protein with the tag on it, and its binding partner will come along with it. And then you can look and see what is bound to it. We're using biochemistry. Using a Western blot. I keep saying biochemistry, but really it's a Western blot, which you've learned about before in class. Another way these affinity tags can be used is that they can be used in a process called immunoprecipitation, wherein you have your protein with this tag on it, and there are antibodies that specifically bind to these tags, so that's an antibody right there. And then you can attach these antibodies to a big heavy bead. I'm not drawing it as big as it really is, but it's this Sephiros bead that's heavy, and since this bead is attached to the antibody and the antibody attaches with high affinity to the affinity tag, when you put this in a centrifuge and spin it, this will pellet down to the bottom of the centrifuge, and whatever is attached to the antibody will also pellet down. So in this case, it would be this protein that you've cloned with its tag and any binding partner that it happens to be bound to. Then you can analyze this pellet with mass spectrometry or a Western blot or some other biochemical means to discover what binding partners are bound to that protein. So there are two ways that you can really separate out these proteins using these affinity tags: through a column or through immunoprecipitation, which is spinning, basically. Okay, let's get back to PQE31 and look at how this his tag works. So I want you to see what it says 6 his here. What this means is within this sequence of the plasmid, there are 6 codons for the amino acid histidine. And in native proteins that you would find in the cell, this is a sequence of amino acids you would never find. It would be very rare or never, I could probably safely say never, find 6 codons of histidine all in a row in a protein. So this contains 6 codons of histidine, 6 codons that encode for the amino acid histidine. Notice that right before that there is an ATG sequence. So this is a start sequence. And so what happens is when you express this, when you put this plasmid into cells and this promoter is activated, where RNA polymerase binds to the promoter here, and we have an inducer that has released the repressor from the lac operon sites, then RNA polymerase will create an mRNA, and that mRNA will contain this start codon, these 6 codons of histidine, and the gene that you have cloned into this multiple cloning site. Those will all be in the mRNA. Of course, in the mRNA, you'll have an AUG instead of an ATG, but then you'll have that 6S histidine, and then you'll have the gene that you cloned into the multiple cloning site. And then notice that afterwards you have stop codons, it's plural. You actually have multiple stop codons. This is going to be important when this mRNA is translated so that the ribosome makes no mistake that it's supposed to stop translating when it gets here. So they have multiple stop codons just to ensure that the translation stops as it should. So when this mRNA is produced and then goes into the ribosome, it will be translated, and when it's translated, of course, we'll have a methionine translated as the first amino acid, and then we'll have 6 histidines in a row here. And then the gene will be expressed, and then there will be a stop codon, and the protein will be spit out from the ribosome. And so you'll have, at the end of this, a protein that's expressed from your gene, and then you'll have those 6 histidines with a methionine, and so you've basically created a protein that has an affinity tag on it. Now there's one thing that I want you to notice and think about briefly as before we move on, and that is, notice that there's a start codon and then 6 histidines all in a row. Remember that mRNAs are read in triplets. In other words, the message of the mRNA is read in 3 bases at a time. So here you have 3 bases, and that encodes for methionine. And here you have 3 times 6, you have 18 bases, each of which encode for a histidine. Now, when you clone in a gene, you need to make sure that this gene is cloned in an orientation that's referred to as in frame. And what this means is that the start codon for your gene should be downstream from these 6 histidines by a number of nucleotides that is a multiple of 3. And that's important because as the ribosome reads the start codon and the 6 histidines, it will set the frame for how this mRNA is read. And so as long as you have a multiple of 3 bases between the start codon of your gene and the last of the histidine codons, then that ribosome will continue to read that gene like it should. But if you had something other than a multiple of 3, let's say you only had 2 bases between the last codon of your histidine and the start codon of your gene, then the ribosome, instead of reading your gene like it should, it would actually read TTA, TTA, and then TG and whatever comes after that, and it would read your gene wrong. Your gene is supposed to be read ATG, not TTA, TG, da da da. So you have to make sure that if you included any flanking sequence in your PCR product, so that when you cloned your gene into PQE31, you have to make sure that there is a multiple of 3 bases between your hist tag and your start codon, so that this mRNA is read in frame. There are a number of different tags that are used, not just histidine. Some of the more common ones that I'm familiar with are called MIC tags, flag tags, HA tags, GST tags. To get these different types of tags onto your protein, you'd have to use other types of plasmids to clone into, not PQE31. PQE31 has a his tag. Now I want to show you briefly how a his tag could be used in a column to purify this protein away from all the other proteins in the cell. So a column that is used to purify his-tagged proteins is called a nickel NTA column. And in these columns there are these, so a column is just a plastic tube that has inside of it some sort of substance that the proteins flow through, kind of like a filter. And as they flow through, if they have an affinity for that substance, they will stick. If not, they will keep flowing through and be collected in a tube underneath the column. And so in these nickel NTA columns, there is a substance called NTA that's attached to nickel, and only things with a histidine tag will have an affinity for them. So NTA is this organic compound that is going to be bound to these beads, cephalose beads. This NTA is a chemical that's bound to some beads that are made out of cephalose, which is just a form of agarose. It's a derivative of seaweed, actually. And these are basically lodged in the bottom of this column. And then this NTA interacts with nickel, which has a positive charge on it. So it's a nickel ion. And it'll actually make this whole column a little bit blue, that nickel will. So this positively charged ion here will have an affinity for negatively charged ions, or negatively charged anything, like a negatively charged amino acid. What we're going to do is take our protein that has this his tag attached to it and put it in a basic solution. And what that will do is cause the histidine to have a negative charge. And then when we put this protein onto this column, because it has that, and it has 6 of these, right? 6 of these negatively charged histidines. That high amount of negative charge in one small area will have a lot of affinity for these nickels. And so that histidine with that negative charge that's attached to your protein will have a lot of affinity for the column. Other proteins have histidines, right? Other proteins in the cell will have histidines, but they won't have 6 histidines. So they may bind to the nickel in this column, but they won't bind with as high of an affinity as our his tag protein, because it has 6 histidines right in a row. So as you pour the cellular lysate over this column, all the proteins that have low or no negative charges will elute out, will flow through the column, and you can collect them or you can just discard them depending on what you want to do. But your protein that has the tag on it will be stuck on the column, along with other proteins that have some negative charge on them. Now what we need to do is get rid of these proteins that are not our target protein, and they don't have this his tag, but they do have some negative charge, and so they've stuck somewhat to that column. What we're going to do to get rid of them, since they aren't bound with a really high affinity, we can actually wash those off using a compound called imidazole, which looks very much like histidine. So it's a structure that's very analogous to histidine. And so basically what happens is if you put imidazole onto this column, anything that's not very tightly bound to the nickel will be replaced by the imidazole, and it'll basically boot off whatever that protein is that's bound somewhat lightly. Some other proteins bound somewhat lightly to that nickel. That imidazole will basically replace that protein being bound to nickel and allow that protein to flow through, so that then the imidazole is bound instead. So it'll outcompete the protein for access to the nickel, and that'll allow the protein to flow through, but the imidazole won't be strong enough, because we're going to add it at low concentrations. It won't be strong enough to remove the protein that has the his tag, and so that will remain bound to the nickel. So then we're going to do a couple washes with this low concentration of imidazole, and that will remove all of the proteins, presumably, except the one that has this histidine tag, which is the one that we cloned into PQE31 that has our target gene expressed. And so that protein then is stuck to that column, so we need to elute it off. And how do you think we're going to elute it off? Well, we're going to add a high concentration of imidazole. And that high concentration of imidazole will be sufficient to break this bond between the nickel and the 6-histidine tag. And that will allow that protein then to be eluted off. And you can collect that in a separate tube. And in that tube, then you should presumably have a fairly pure fraction or a pure sample of your protein and whatever binding partners it's bound to. And you to collect that in a tube, and that would be your what we call eluent, which would have your protein, your tagged protein of interest. Then you could go on to do biochemistry with that eluent to see what proteins also were bound to your protein of interest. There's another type of column that we're going to use to purify our protein in class, but it does not use the his tag. It's called HIC, or hydrophobic interaction chromatography. These HIC columns use the hydrophobicity of proteins to separate different proteins from each other. And so how it works is, if you think about your cell, the cell has lots of different proteins in there, and some of them are more or less some of them are, these proteins vary in their hydrophobicity. Some are a little more hydrophobic than others. And what an HIC column does is take advantage of that difference in order to separate proteins from each other. So if we have our protein of interest that we've cloned into PQE31 and it has this histidine tag, well, this HIC column is not going to use this histidine tag to purify this protein, but it's going to actually use the hydrophobicity of the protein itself to separate it from other proteins. Now, what if you have another protein in the cell that has the exact same hydrophobicity as your protein? Well, then you won't be able to separate them from each other. So what do we mean by hydrophobicity? When you learned about amino acids back in Bio 180 or in your biochemistry class, you learned that certain amino acids are more polar than others, and there is a whole group of amino acids that are nonpolar. These nonpolar amino acids tend to, when the protein folds, collapse. They tend to collapse into the center of the protein. But every once in a while, you'll have some of those nonpolar amino acids be exposed on the outside. So typically, they'll collapse into the center of the protein. But sometimes you'll have these nonpolar amino acids also found on the outside of the protein. And depending on the amount of these nonpolar amino acids you have exposed, this protein would be considered more or less hydrophobic because it has more or fewer exposed nonpolar amino acids. In an HIC column, we have beads that are nonpolar in the column. So they're not the cephalose beads that are attached to the nickel to the NTA. They're a different kind of bead. They're white in color, and they're relatively hydrophobic. And so if you have a protein that is highly hydrophobic, it will bind very well to these beads in an aqueous solution in water. Because of course, hydrophobic things want to get out of water, and so if they are in water and they come close to each other, they will associate together to try to escape water because they're hydrophobic. And so if you put something that's very hydrophobic over these beads, it will have a lot of affinity for these hydrophobic beads. Like attracts like in the case of hydrophobicity. And so what we can do is we can separate proteins into different tubes based on their hydrophobicity. And this is how it works. What we're going to do is break open our cell and remove all of the proteins and put them in a high salt solution. A high salt solution. So all our proteins will be in there. In a high salt solution, because they're in with a lot of salt, there's a lot of charged ions in the water. If there are hydrophobic proteins or proteins with hydrophobic patches on their exterior, that salt makes them even more hydrophobic relatively, because the charges make them even more likely to bind to other hydrophobic surfaces, more than just water would. And so if you have a protein with some hydrophobic patches here, let's say you have a protein with 1, 2, 3 hydrophobic patches, and you have another protein that has just 2 hydrophobic patches. Well, in salt, both of these, if you had salt with a lot of ions floating around in the water, then these hydrophobic proteins are not going to like this at all. They will not like being around these ions, these charged molecules, and so they would rather bind to these hydrophobic beads. And so with a high salt concentration, all proteins will probably bind to these beads because they all will have, to some degree, some hydrophobic patches on their surface, and this high salt will be sufficient to make all of them want to bind to these hydrophobic beads. So once they're all bound to these hydrophobic beads in that high salt concentration, then what we're going to do is add little aliquots of lower and lower salt concentrations to this column, and with each lowered amount of salt that we add, we will elute out some of the proteins into a tube. So, for example, this guy here that only has 2 hydrophobic patches, it does not have as much affinity as this one does with 3. And so if we just drop the salt concentration a little bit, this one won't stick this one will be the first to leave the column and drip out into our tube. Then if we add another solution of salt that's a little bit lower in salt concentration, then that may be enough to allow this guy to be released from the hydrophobic column, and we can release that into a second tube. And so what you can do is separate these 2 proteins that have different number or different amount of hydrophobicity on their surface by gradually decreasing the salt concentration. Those that have fewer hydrophobic patches will be released first, and you can collect them into one tube, and those that have more hydrophobic patches will be released next, and you collect those in a second tube. And then the ones that are most hydrophobic will be released when you add pretty much pure water. They'll be released from the hydrophobic column and then flow out into another tube. And so by the end of your fractionation or your separation of these different proteins, you'll have various tubes, each of which have proteins of different hydrophobicity. And what you're doing is purifying your protein of interest from the other proteins in the cell based on their hydrophobicity. Again, if you have another protein that has the exact same hydrophobicity as your protein of interest, then they may end up in the same tube together. Now what you could do with that tube is you could take it and put it over another column, and this time change the salt concentration by even smaller gradations, in the hopes that the protein that you want and the protein that you accidentally collected have just slightly different hydrophobicity, and what you can separate one from the other using smaller differences in salt concentration. So this is called an HIC column. It doesn't use an affinity tag, it just uses the natural hydrophobicity of proteins, but it's another way that we're going to, in class, separate our protein from the other proteins in the cell. Which one of these two processes will give you a more pure sample of protein, it would be the one using the affinity tag. This one's going to give you a much more pure sample of protein, of your target protein in the eluent, than this would over here, the HIC column.